computer, and we're going to share our screen. We'll pick up our anaerobes, and here we go. All right. So this is I want to say is it chapter 41 or chapter 40? The overview, I think it's 40.
It's 40.
It was 40 and 41 for these two. Yep. So the anaerobic bacteriology in 40, and then we have 41 on Thursday. So these are anaerobes. You've been waiting all semester to get to anaerobic organisms. So the organisms, they fail. That's the way we start. They fail to grow in the presence of air or molecular oxygen. So these are the ones that we have to set up a different way. We have to set up a different way of growing these, because we can't just do our normal, streak them on a plate, put them in the incubator. We can't do the increase the CO2 level with a candle jar. Okay, so we're gonna have to knock out the oxygen totally. So in reality, it says anaerobes exhibit different degrees of oxygen tolerance. Some are able to flourish and grow even though their atmospheric conditions are not optimal. So we're going to go through the classification of anaerobes. So this is chapter 40. So we do have aerotolerant anaerobes. They grow best in the absence of oxygen, but are still able to produce colonies in ambient air when they're cultured on fresh media and the inoculum is heavy. Okay, so we do have that. We have moderately obligate anaerobes, and we know that obligate means what? Obligate, we've hit on this since day one. Obligate, that's just facultative. Obligate is strict. Yes, that's the word we like to do. So we're moderately strict. That doesn't sound like we're extremely strict. But moderately obligate means we don't grow when exposed to oxygen unless the time of exposure is very brief. These bacteria can tolerate a limited amount of oxygen toxicity. Most of the anaerobe organisms isolated in the lab belong to this group. Okay, so that's why we're talking more about obligate anaerobes. Strict obligate anaerobes are extremely sensitive to oxygen. That means if they get a hint of the oxygen, they cannot handle it, and thus they're not going to grow. So they actually die. So techniques for collection, processing, and isolation of these strict obligates requires an oxygen-free environment and careful attention to detail. So I think to one of my case studies that's in the clinical round right now that the clinical students are doing, one of those talks about aspirating an anaerobe, and it says, and this is helpful because you will answer correctly if you remember this, it says, how would you collect a fluid anaerobe, and why would you do it that way? And it's a needle aspirate because it keeps it in an anaerobic environment. The needle actually goes into your body where it's swollen, pulls out the fluid, and then contains it into a syringe that's pretty oxygen free if you didn't open it in the air and pull air in there first. Okay, so we can keep it that way. So those are definitely a great way to collect an anaerobe from the body. Because if we get exposure to oxygen, then the organism dies. So what makes oxygen sensitivity? Well, hopefully we've touched on it a little bit, but it's two factors. There's one factor of production of a toxic product, such as hydrogen peroxide and superoxide ions. So those are the two things that these organisms cannot prevent from happening through the interaction of oxygen with the bacteria themselves. So that's the problem. If they're oxygen sensitive, they're not going to handle oxygen peroxide. you know, which we're fixing to see, that there are certain ways organisms can prevent those hydrogen peroxide and superoxides from killing them. Our aerobics, ones we've been working with all semester, some of them possess enzymes that degrade hydrogen peroxide. Wow, that's something. And superoxide ions. So some anaerobes possess catalase and some superoxide dismutase, but strict anaerobes have very low or undetectable levels of dismutase. So SOD is superoxide dismutase enzyme. We see this oxygen molecule here. This is the hydrogen peroxide and water. And then we see catalase enzyme that takes care of the hydrogen peroxide and turns it into water and oxygen. Thus we get our what? We get our bubbles, right? When we do the catalase test. So that enzyme is absent from our strict anaerobes. You've seen that already, right? You've seen catalase work. A second factor associated with oxygen sensitivity has to do with oxidation reduction, redox potential. Ooh. Remember, I think we mentioned redox, like we love redox, right? Didn't we talk about how we were going to come to redox eventually? And y'all were going to reach back and get your chemistry knowledge. The reason I sent you to chemistry class, like Chem 1 and Chem 2 and organic, right? The reason you had to go into those was so we could at least understand redox. And Heather's online, so Heather's already been through clinical chemistry, right, Heather? Somebody told me to check my Zoom people today if they weren't turning on their cameras. All right, Heather's been through clinical chemistry. So Heather, we're going to rely on you to help us with the redox potential because a lot of our clinical chemistry is based on redox. The growth and metabolism of anaerobes requires low redox potentials. The presence of oxygen results in high oxidation reduction potentials. So let's read this little bottom here. Low redox potentials keep reversible components of various enzymes in their reduced form rather than their inactive oxidizable form, preventing structural damage. Okay, so what we see with anaerobes is what? Low redox potential. What we see with anaerobes is the presence of oxygen results in high oxidation potentials. Okay, so the growth and metabolism of anaerobes requires, did I say that wrong again? Let's say it one more time. The growth and metabolism of anaerobes requires a low redox potential. The low redox potential keeps reversible components of various enzymes in their reduced form rather than their inactivated oxidizable form, preventing structural damage. Let's put those two together. I'll give you a chance to digest that. We got to get that straight. So if I said, what does an anaerobe need? You would tell me what? Low redox potential, correct. Yes. And what is low redox potential? It's a state of the organism that keeps reversible components in their reduced form rather than their inactive oxidizable form, that thus preventing structural damage from the oxidizable form. So how do we collect anaerobic specimens? Proper collection, avoid contamination with commensal microbiota, which may overgrow the anaerobe. Avoid exposure to air, which affects the ability to recover and cultivate anaerobes. So let's think about how we get womb cultures, right? Is that the culture we think would be an anaerobe? somebody had a wound, whether it be an abscess or whether it be an internal something going on, maybe they find it during surgery, right? Some fluid from surgery they want us to do a culture of. We're going to do it both ways, right? So when we get those kind of fluids, we'll do it aerobically, all right? We'll put it on blood agar, we'll put it on McConkey, we'll put it on chocolate, we'll put it on PEA, mannitol salt, we'll put it in thioglycolate broth, okay? We'll do that. And then we'll also streak out an anaerobe blood agar plate. And we'll put that in an anaerobic environment. So if that culture had been exposed to, if it was strictly, right, if it was a strict anaerobe, well, that wouldn't be good, would it? Because any exposure to oxygen, if we took the swab and waved it in the air, we're exposing it to oxygen, right? Right? So that was the difference. What was most of our clinically important anaerobes? What are they? Moderately, right? Moderately obligate, where they can handle a little bit of exposure. So what we're going to do is we're going to put them in their environment. So the reason we put an anaerobe environment with the bag, put that plate in there, and put a gas pack in there to alleviate all the oxygen that's in the bag, right? Replacing it with a different gas. Then we can set up for that, what? Anaerobic growth. So we may look at our plates and we see positive growth. We see aerobes, right? We may see staph, we may see strep, see E. coli, we see pseudomonas, we see all those growing out of the wound. But we're going to go to that anaerobe plate where those shouldn't be growing, okay? And hopefully we will see that we get our anaerobe growing. So this is really tricky when it comes to the surveys, right? We talk about proficiency surveys and our API surveys that get sent to the lab. They won't tell you they're looking for an anaerobe. They'll say culture it. And you set it up like you do any other culture, and then there might be a hint of an anaerobe, which means there's only growth on the anaerobic environmental plate that's not seen elsewhere, but there's other growth, okay? So you gotta be careful as to not missing that anaerobe growth, especially on that. And we definitely wanna know if we have an anaerobe. So you don't wanna miss it, it's real easy to miss. So avoiding contamination with commensal microbiota, so we're gonna try to eliminate microbiota, and the way we do that is put it in an anaerobic environment. So what makes it unacceptable? Let's check this. So then this gets tricky because this becomes our potential hotspots for interprofessional discommunication or miscommunication or hard feelings or ticking people off, in other words, right? When we decide it's unacceptable, that's your decision. That doesn't always fly very well when you call and say, we're rejecting your specimen you just collected for us, because it's collected incorrectly. Unacceptable specimens for an anaerobe would be oral and nasal cavity specimens, okay, unless we're looking for actinomyces. Gastrointestinal tract specimens, except for C. diff cultures. And just to give you a little hint, we're working on a project maybe coming up soon where we're going to be looking for C. diff. We may be looking for volunteers, professional extra point volunteers that want to be part of our little study where we may be testing out a new white, okay? So we're maybe doing this sooner than later, where we may need human subjects for disinfection. So C. diff comes up. So C. diff is an anaerobe, and it's from the GI tract. It's usually hospital acquired, and it's usually spread. So we're working on how do we want that to be prevented, that spread. Genital urinary tract specimens, except if it's a suprapubic urine, which means we go past anywhere oxygen would be present. Material from ulcers and open skin lesions, of course, would be unacceptable. Acceptable, which we just talked about, that little case study that I've got in my clinical list, is body fluid, right? Cerebral spinal fluid, pleural fluid, joint fluid, pericardial fluid, peritoneal fluid. Those are all acceptable. Okay? And we just need to make sure we collect those as anaerobically as we can with a needle. Abscesses or tissue biopsies, lower respiratory tract, so like a lung aspirate, sulfur granules from draining fistulas. Y'all know what a fistula is? Anybody? Fistula? Never heard of that? Or has anybody ever had a fistula? It's like your body decides that it needs an extra exit from some part. So this little thing grows out like a little channel, like a little connecting tube. And it finds an exit somewhere that it can get it out. And you have a fistula form where there's an exit, usually due to an infection. Okay, your body's pretty neat. But that would be the sulfur granules from that, the draining fistulas. Blood and bone marrow are acceptable. Because we know when we do blood culture collections, we put one where? In an anaerobic and an aerobic. We have 2 bottles. That's why we have 2 bottles for blood cultures. Have y'all collected blood cultures in what's that first class called again? Principles. Did y'all do that? Y'all didn't collect blood cultures and principles? You might need to do that then, huh? Y'all didn't go over phlebotomy, and when you use a butterfly, there's about that much oxygen, so you want to put a butterfly in the aerobic first before you put the blood in the anaerobic. Y'all didn't go over that principle? Okay, just seeing where we are. Okay. Y'all did decontaminate the arm maybe with hexokinase or what's the little popper? Iodine versus, no, y'all didn't do that either? Okay. All right. Clues to anaerobic infections. Let's look at those. Foul odor, of course, yes. And believe me, the little gas pack, when you open up your anaerobic gas pack, so you don't want to open that until you're ready to take the plate out. So the reason they're clear and the reason that you don't open those is so you can actually see the growth. So I get really irritated with my anaerobic bag when somebody puts the gas pack pouch in front of the window of the plate that I want to look at the growth. And I have to try to move that with my hand without opening the bag. You don't want to open the bag and let the gas out, let oxygen in. That's not good. So you want to seal these bags and keep them sealed. So you want to be able to read through the plate, okay, read through the plastic bag to see if there's any growth on the blood agar inside that anaerobic plate. So we have this. We're going to set up these. This will be fun. We have these gas packs. Infections in a close proximity to a mucosal surface. Infections following human or animal bite. So anytime there is a puncture wound in the skin that goes deep, we want to make sure we're being maybe an anaerobic growth. If it festers after the bite or it festers after it's sewed up. I think we were talking about this the other day about sewing up somebody's wound too soon before they get it cleaned out, because that anaerobe gets in that wound. You close it up with stitching, and then all of a sudden it starts to swell and gas is produced and it starts to smell, and you're like, oh my gosh, what did I do? You just gave the anaerobe an environment that it wanted, which by sewing it up. So some wounds are left open for that, so they don't set up an anaerobe growth. We may get anaerobes if we've had previous antibiotic therapy, and we're going to talk about this with our aminoglycosides, kanamycin, gentamicin, tobramycin, and neomycin. Black discolorization of blood-containing exudates, sulfur granules in the discharge. We have unique morphology on the gram stain. We have fusiform gram-negative rods or large gram-positive rods with spores. So if you didn't know, C. diff has spores. So we're going to be talking about spores in this anaerobe environment. And then we do our gram stain, and they do not grow. Organisms are seen on gram stain, but they're not growing aerobically. and then growth of the anaerobe portion of the liquid or semi-solid media. And that goes back to our thioglycolate gel, or broth, right? Usually we introduce that with our swab that's been taken from the patient. We put that down in the growth, in the media, and pull it back out. So what they're saying is the farther away from the oxygen at the top of that, it starts to grow down here at the bottom, you may have an anaerobe. So we'll look at those, and you see the growth at the bottom of the tube instead of the top, or through the whole thing. So caution, caution, due to graphic nature of these program. We see an ulcerated lesion, debrided, okay, and hopefully they debride that before they bring you a culture of it. Subdural empyema, a mixed infection. I think I can see some here, I can see some there. It's not really focused, but hopefully you can see your gram stain working and bringing in some gram positives and gram negatives. All right, so growth requirements. Well, I love chapter 40. It's got a lot of good stuff. And it's not that long. Packed with stuff, though. All right, a lot of charts. Heman. Where have we seen Heman before? Yes. So we know chocolate might be a good spot, right? Because Heman has been seen to show that. Vitamin K enrichment. Sodium bicarbonate. CO2 stimulates growth and spore formations of Clostridia. Amino acids, L-cysteine, cysteine for sulfur-containing amino acid dependent specimen. Yeast extract and blood. Reducing agents to absorb the oxygen and lower the redox potential. Makes sense because we just talked about how we want that. Pre-reduced anaerobically sterilized media, sterilized and packaged anaerobically. So you can actually get a plate that's sitting there already anaerobically. Non-selected blood agar. And this is what we see most in the lab. We don't see this pre-reduced anaerobically sterilized. What we normally see is non-selected blood agar. Anaerobic blood agar plates differ in the growth supplements from the blood agar intended for cultivation of aerobes. And the way they do that is the addition of the vitamin K and the Heman. So if we have a plate, right, which it looks just like our regular blood agar plate, but if you look on there, it will say anaerobic blood agar plate. And that change is vitamin K and Heman added to that. Okay. They're a little darker, but they still look like blood agar plates if you pick them up. Okay. At this day and time, we know we're running, what I'm hearing a lot is shortage of blood agar plates. Not all hospitals are able to get them. They're back ordered. They're having trouble supplying just that simple of a thing. And you may or may not have the anaerobic plate for your wound culture. But hopefully you do, because that's key. You want to go ahead and put that plate with the anaerobic environment, double it up. So the substitute, next best thing is to just use a regular blood agar plate, put it in the anaerobic environment. It won't be as good as it could be, because we want to definitely have the vitamin K and Heman added to that anaerobic plate. But we do have those, and if you took them side by side, you'd be able to pick up one and not notice you had which one you had. So be careful there when you're in clinical. We do have selective media. We have selective media for different anaerobes. And right now I'm not worried that you're seeing these anaerobe names because we're going to come back to those. So I'm not really, you know, we got another lecture coming up Thursday where we'll put the names, start putting the names to these. But right now we're just kind of getting the idea of what's important. But you can, you know, Bacteroides, you can put it there. That one needs a kanamycin and a vancomycin blood agar plate. Probably not going to find that in your normal, like, hey, hospital lab, do you have that kanamycin, vancomycin plate? I think I got Bacteroides here. That's not the way it's going to work, okay? We do have anaerobic PEA, okay, which inhibits that growth of the microbiota of the positives, right? Anaerobic gram-negative bacteria and anaerobic gram-positive bacteria are not affected by PEA. So we would see that. We would see growth there. So that's one of our selective medias. Bacteroides bioesculin agar for Bacteroides fragilis group. And then at the last one, we see a cycloserine cefoxitin fructose CCF agar for C. diff. So we have some very selective, but you know, this would be, this is when you would hope, well, I say this, if we could keep plates longer than usual, you know, usually these things may go out in, you might get a box in and you may see a 6-month expiration date. Okay. You may not get an anaerobe for 6 months at the hospital. You may, depending on how good the surgery is, or if there's a potential contamination during surgery, or they don't sterilize their instruments very well. You could definitely see that. But that's what's, when I say we don't have everything right there, that's because we just can't keep ordering over and over and say we've got all these sitting here on the in the fridge, okay? Because with clinical lab, unlike your student lab downstairs, once they expire, they're supposed to be thrown out. So it just becomes a demand. If there's a demand for it, sure. But the demand's usually not that high for anaerobes. But those are some, like I say, these are our toys. And it's nice to have the toys available if you do have a chance to keep this. But I can tell you, I've never seen a kanamycin, vancomycin blood agar plate. I don't know if I've ever seen an anaerobic PEA. I have seen PEA, but I don't know an anaerobic PEA plate. But usually the one I've told you about was the blood agar with the hemin and the vitamin K. I have seen that one. That one should be there. Thioglycolate broth, that's there. Okay, liquid media. So we do use that one, and that's usually with our wound cultures. We set that up. We put the swab in that they send us a swab from the wound. If we just have liquid, you know, if it's CSF or if it's from an exudate, we could just do that, add it to the thioglycolate broth. Again, both are supplemented with what? Hemin and vitamin K. Okay, so to encourage anaerobic growth. And then with thioglycolate broth, we see sodium thiolate acts as a reducing agent, and 0.05% agar controls the oxygen diffusion. So definitely the thioglycolate is great for anaerobe growth. So if you don't have that anaerobe blood plate, definitely want to have thioglycolate broth. And most labs I've been to have that. They order a case of those and they last a lot longer than the agar plate. And you can definitely use those as part of your setup. So that's what they look like. Okay, they come like that. And this is what I do right here. I stick the swab in at the end and I leave the swab. Instead of throwing it in the biohazard, I just go ahead and leave it in the thioglycolate. And I snap off the top. And I want to make sure that that top is not contaminating anything. So I want to make sure that if I feel like somebody's touched that top of the stick, that's the part where I break. And then I put it in the incubator overnight and see what it's looking like the next day. It'll cloud up. If there's growth, if it's anaerobic, it'll grow down here, okay, away from the top of the broth. So we have our anaerobe jars, okay? These are different than candle jars. Don't make that confusion that, oh yeah, that's anaerobe growth. Mr. Director always lit a candle and put it in there, right? No, these are the gas packs. So we're gonna have to really, usually you can use the same one like I've used for that, but usually you have a little holder where the gas pack slides in. You can put more than one plate, of course, you can stack them up. But what we're going to do is we have a palladium catalyst, okay, that catalyzes hydrogen and oxygen into water. We can also use a hydrogen carbon dioxide generator. The reaction of citric acid and sodium carbonate, CO3, provides CO2. The reaction of sodium borohydride and water produces hydrogen, which combines with the oxygen to form water, eliminating water from the atmosphere. We can do an evacuation replacement. The jar is evacuated using a vacuum and then filled with anaerobic gas mixture. That's extreme, but that can be used, right? So we're just trying to get what? We're trying to get oxygen out of the atmosphere, whatever way we do that. Okay, that's that. You've seen this downstairs, and you saw two little valves here. This looks old school, right? Put your stuff in there and you suck out all the oxygen and replace it with hydrogen gas. Anaerobe jar here. Okay, and we can use a bio bag, and this is where I usually see, this is what we use today. I've got boxes of bio bags downstairs. And we'll try this. We've got some anaerobes we'll try to get growing. The bio bag is an anaerobic culture system. The components are the same as the anaerobic jar. Clear plastic oxygen impermeable bag allows inspection. We've already talked about that. And y'all are going to remember that and not put your little pouch in there over my window so I can't see what's growing. You're going to put it on the back side of the culture plate, right? Where you wrote your name and all that. Right? Not going to make that mistake. So we don't have to open them. We have anaerobic glove boxes. I've yet to see that. But I'm digging downstairs, so I'm in the closet some, and sometimes I find stuff. I did find an old composite picture. Just thought I'd throw that in. We'll kind of break the anaerobic. Does anybody know that like in 1989, we had a health administrator program here, something like that, health sciences administrator? Yeah, so I found the composite today. I brought it out. We're going to send it over to the dean's office and see if she's interested in seeing this. Had about eight students on it. Okay. I don't know if it was a what we do now, which is health sciences major, right? We thought that was like just created, and maybe we already had one, like health administrator. Sounds like a health science major. Anyway, that was in our closet in 117. Found that today. I don't know if I ever told you, but I found a shovel the other day. Did I tell you about the shovel? No? Yeah, I found a shovel. It was on top of the shelves in the 117. And the shovel was used for the groundbreaking ceremony of the Reynolds Building renovation in 2007. It said keep. And I'm like, okay. So I gave it to the dean. Brand new shovel. It's a great shovel. You know how they take the picture, everybody's out front, you know, with a brand new shovel in their hand. And anyway, so we don't know what all's in 17. It's a fascinating room. This would be our glove box. We do not have one of those. Sorry, you won't get to work with the glove box and keep it in an anaerobic and working environment. All right. So I think we're finally through with like what we're doing, and we're finally going to get into some organisms. Y'all ready? Y'all been past ready, right? Y'all like that kind of stuff? Like this is like when somebody says, well, you know how to set up something anaerobically, and go, yeah, I think Mr. Director spent about a half hour on that one day in class. You're not going to forget it, right? So our anaerobic spore-forming gram-positive rod. That's where we are. Is this on our list? Let's look here. Let's see if we can find it on our handy dandy sheet. Let's see. We got gram-positive anaerobes. Do we have a section for anaerobes? I don't know.
Yeah, it's in the top left, the second one down.
Top left, second one down. Rods. Okay. So that's them over there, all these, does it just say anaerobes on it or what?
The little box underneath the Clostridium says anaerobes.
Oh,
Yeah.
okay. Underneath the Clostridium, a little box, anaerobes boxcar shape. Yes. Okay, so we got still got some help. The big thing, we're with Clostridium. Okay. Clostridium species, we see that. We have Tetnia, we have Botulinum, we have Perfringens, and we have Difficile, which we've got to talk about how that name changed a little bit. Catalase, negative, right? If it was catalase positive, what would we say? It wouldn't be an anaerobe, right? It would be able to handle the oxygen, right? So the catalase is very important. We touched on it from the beginning. We're on our Clostridiums now, and spores. Terminal spores, or subterminal spores, or centrally located spores. So located in the extreme end, it looks like a tennis racket for Clostridium. Subterminal spore located off center, but below the far end of the cell, and we have centrally located. So there we are. Terminal spore, looks like a tennis racket. I don't know. It definitely looks like a badminton racket. Y'all see it? Would you miss it if I showed it to you again? Would you tell me that's the spore, right? That right there is the spore. Okay, right here, right there. Subterminal spores, can you find those? All right, so they're not like a tennis racket, but you can still see these. See these clear openings in the rod itself. This looks like a needle to me. Looks like where you take the thread and thread the eye of the needle. That's the way I put this one, subterminal spore. Who's going gram positive rod or gram negative? Who's going, you gonna change on me? Again, look. I see pink, I see red. What's my first impression? There's a lot of purple in there. So Clostridium are large gram-positive rods. So remember what I told you, when we do gram stains, if we see gram-positive, we don't like that, okay? We don't like that at all because we may or may not be able to grow them, okay? So that's why they said, look at the gram stain. If you look at the gram stain, but you don't see any growth, right? And gram staining from a wound, how do we do that? Let's start there. I think this is important because this can be confusing because some people will say as soon as you get a wound, you need to gram stain it. How do you do that? You have no growth yet, right? So you're going to take that swab, you're going to smear it across your slide, and you're going to gram stain it. That's how you do a gram stain before you get growth. Take your culture swab, smear it on there, just like you would if you'd have picked it up off the plate, because they're coming from a wound. So if it's growing there, it's there. And then if you come back the next day and you look at your plates and there's nothing there, that's that concept of there's gram stain, no growth, right? That's how that happened. I don't know if y'all were confused by that when I said it earlier, but that's how you do it. You take that culture and go ahead and gram stain it, and you'll see it if it's there. You'll find it. It's not like it's not going to be there. It doesn't need a day to grow out of the wound. It's growing there already. What we're trying to do is transfer it over to growth plates that don't have anything on it already. That's the day we need to grow past that. But we can gram stain. Somebody swipes a big sore on a person and brings it in and says, hey, and they go, this is from a wound or an abscess and you need to gram stain it. We need to know. That's where you might see the gram-positive large rod with these terminal spores. Okay? Don't miss that. Because I don't want you to say, Mr. Director said we always took it off the growth plate the next day, so I'm not doing a gram stain today. We're going to wait till tomorrow, all right? And then there's no growth, and you go, look, your culture's negative. Hey, aren't we good? But then the patient's sitting there suffering with this festering wound that just keeps being messy.
I have a question, Mr. Rector.
You not need to know that they're parallel sides. just spores?
Parallel sides.
Because on ours, it says large gram-positive rods with parallel sides.
Sure. I mean, that's
Okay.
just that. Just big old rods. Parallel just means they're doing that. Long, cylindrical, long. Yes. All right. Aerotolerant species may grow on a freshly prepared aerobic blood agar plate and could be confused with bacillus species.
There was a
except
question
they're
on
catalase
Zoom.
negative. Ha, that sounds like a test question if I've ever seen one. How
There
would
was
you
a
not-
question on Zoom.
How would you not confuse a bacillus, because we know bacillus is not an anaerobe, right? With an anaerobe. And bacillus, is that a gram-positive rod? Of course it is, but it's catalase positive versus catalase negative. I think you are good to go. It's because we've already covered this. You guys are already on top of catalase.
Can you hear us, Mr. Rector?
So we'll start here. Clostridium tetania.
Mr. Rector,
I don't think he can hear us.
So we were just talking about this the other day, I think, weren't we, about the rusty nail? I think we brought it up in lab. Somebody did about the rusty nail, stepping on the rusty nail, and that's the only way you can get tetanus, right? Not really, right? Habitat, soil, and feces of humans and other animals.
Mr.
Yes,
I think Heather's trying to say something in the chat.
I'm sorry, Heather, you need to unmute. Or she.
Well, she was unmuted a second ago, but I don't know.
There she is.
Shirag
What you
had,
got?
Shirag had a question, but you didn't hear
Hello,
her.
Heather.
Okay. Can you hear us now, Mr. Rector?
I don't know.
Yeah, she just sent me a text that said, hey, he can't hear us on Zoom.
Yeah, he can hear us on Zoom.
Oh.
Somebody turned down the volume for some reason.
Okay. Can you hear us now?
Yes, you're loud and clear now. Sorry, somebody turned down Zoom volume for some reason.
You're fine. I just had a question about when you said, how do we collect the culture and how do we put it on the gram stain? So
Yes.
we don't actually pick it from the plate, we pick it from the patient.
You're actually going to take, yes, the nurse will give you a swab from the wound and say, please culture the wound. So you're going to take that swab that they give you, and you would normally, you're going to streak a plate with that. You're going to load your plate with the swab, right? And then streak it for isolation. You would also take that swab and put it on a class slide to be stained for gram stain.
Okay, so we plate it and put it on the slide.
Yes. And I
Okay.
would probably put it on my slide first, just to make sure I got as much on the slide to begin with.
Thank you so much.
Okay, no problem. Sorry we didn't have your volume turned up. Sorry. Thank you, Heather, for alerting us, because I don't have the chat box up with me when I'm sharing my screen. Sorry. I have
No problem.
I have this over here. I see Lauren's put up a ghost of some kind. A ghost. All right. So let's go back to Clostridium tetani. Soil and feces, the spores remain viable for decades. That's the problem, right? That's what we're talking about here. So we're talking about spores, and we're talking about, is the spore the problem that's picked up? So transmission is the organism enters the body through a wound contaminated with dirt and feces. So that's what we think about a rusty nail. It's nothing to do with rust, it's just a puncture wound. Like, so anything that punctures your skin, walking through the woods or out running around with no shoes on, right? That's getting into an anaerobic area. So the farther away it goes into your skin, away from the opening, and it closes up, and then that's where we set up that environment. So not necessarily a rusty nail, but just some kind of puncturing device. So that spore germinates into the vegetative cells in the tissue where the redox potential is low, right? Because it's a puncture. And due to other bacteria and necrotic tissue, toxins are produced as this vegetative cell grows and reproduces. and the toxins are absorbed into the bloodstream. And that's when we start to have tetanus, right? That's when we have our lockjaw and muscle contraction. Hopefully you've seen this before. So there is our tennis racket, right? Our tennis racket with the spore, terminal end of the spore on our gram-positive. Like how many of you before today would have probably stained that and go, I don't know what we're looking at, right? That looks like something out of, not out of my micro book. That looks weird. That must be some kind of artifact. I'm not going to say that's important, right? Now you've seen it, so hopefully you won't miss it. So the tetanus toxin is not good. It says FYI, but I think anything we cover is always good to know. Tetanus spasm is a highly toxic spasmogenic neurotoxin. That's what causes the tetanus. Tetanus is characterized by painful muscle spasm leading to respiratory failure. 40% of all cases lead to death. So that should scare you. It does. That's why we like to have tetanus vaccinations. All right. Tetanolysin, a hemolytic toxin, may also be involved in the symptoms of tetanus. The mode of action is the tetanus toxin binds irreversibly to nerves, where it stops the affected neurons from releasing inhibitory neurotransmitters. Thus, this inhibitory neurotransmitter normally inhibits the alpha motor neuron. I don't know we're going to get into neurology, but we are. With diminished inhibition, the resting firing rate of the alpha motor neurons increases, producing a rigidity, unopposed muscular contraction, or a spasm. This toxin spreads through the lymph and blood, through the tissue to the peripheral nerves, and what we see is lockjaw, what is known as lockjaw or trismus, which is muscle spasms. Was anybody ever scared of lockjaw when you were a kid? Did your parents scare you with this? Like, I don't know, you better wear some shoes, you're gonna get tetanus. No. And then have you ever heard somebody having to have their jaw broke because of lockjaw? Anybody heard that story too? No? No, okay. All right, well, early symptoms of lockjaw are tension, cramps, and twitching around the wound, dysphagia, stiff neck and jaw, headache, backache, irritability, tachycardia, and then that leads into the later symptoms of lockjaw, trismus, spasms of the jaw, followed by spasticity of the neck, trunk, and limbs. Okay, so it's that, like that, right? It's that grit, like you're gritting your teeth, you can't open your mouth, but everything's locking up, everything is contracting around your mouth. That is the classic there of tetanus. That is the arching of the back. See that in old pictures from back in the older days. It does occur to infants without protective passive immunity from mom, because mom was not immune. It usually occurs to infections of an unhealed umbilical stump. So we never thought cleaning the umbilical was that important. And I know none of y'all have kids. Heather, help us with this. Right? Just kind of like at least dab it a little bit with a little alcohol pad every once in a while until it actually, heals up and falls off, right? I don't know if y'all knew that.
Yeah, you have to keep it clean.
Yes. Yes, this is, maybe we'll take that a little more serious now, right? But hopefully you were giving passive immunity to tetanus to your children because you were vaccinated against it forever and ever and ever. Every 10 years, right?
Yes.
Generalized tetanus, most common type, about 80%, starting with the trismus and then going to lockjaw, followed by stiffness of neck, rigidity. What is the therapy? We got to neutralize the toxin. We're going to give antitoxin, relieving that spasm and maintaining an open airway, preventing further toxin production by removing the infected tissue. Oh, that sounds horrible. We got to go in and surgically remove the tissue that's infected. All right, so that is tetanus. Anybody have questions about our Clostridium tetani? So does anybody know this one?
So Botox isn't, or no?
Botulism, which it might be Botox. Yeah. All right, some people do this. Clostridium botulinum. I'm thinking botulism. You might be thinking Botox injections. Let's see who wins. Habitat, Clostridium botulinum. Soil and water, fresh or marine, it doesn't matter. The toxin, most powerful toxin known to man. More than 1,000 times more potent than a rattlesnake venom. Mode of action, it acts as a neuromuscular junction of peripheral nerves blocking the release of acetylcholine. As a result, no impulse can be passed across the neuromuscular synapse. Without muscle contraction, the patient is paralyzed. Imagine, all right? Again, terminal spore, tennis racket, badminton racket, whichever one you prefer. Even more. Closer view. We can grow this one on egg yolk agar. Lipase test positive with the sheen. The lipase positive, but we're using egg yolk agar. It's a foodborne botulism. So we see 3 clinical forms. The first one we're going to talk about is home canning of the food. It doesn't have to be home canning, but usually that's where we see contamination, right? Because what we do is the source is inadequately processed home canned foods. So hopefully you were aware of that. If the food is contaminated with Clostridium botulinum spores, it has not been heated adequately, the vegetative cells germinate, producing the toxin. If the food is not then thoroughly cooked, the toxin is ingested, causing nausea, vomiting, and diarrhea. And then we end up with neurological symptoms appear 18 to 36 hours later. Then we have paralysis of our respiratory muscles, and we can't breathe and we die. Okay? That's just a horrible way, I would think, to be enjoying a nice homemade food that your grandmother just canned for you, right? And boom, right? So you always want to make sure you don't forget to use the heat or at least cook it. And there we are, our contaminated jalapeno peppers. Infant botulism follows ingestion of spores which germinate in the infant's colon. How in the world would that happen? The source is usually food. The immediate environment is contaminated with spores, and all cases occur in infants less than 1 year old, where intestinal microbiota has not been firmly established. The organism proliferates, producing a toxin which is absorbed into the bloodstream, causing symptoms of botulism. Honey, the honey we love, the reason we don't give it to infants, because honey is commonly implicated and has been recommended that we do not give it to children 1 year or younger. So do not feed honey to your babies.
I think they said Cairo syrup as well.
Sure it did, yes. Good call. We have wound botulism. Follows an implantation of the spore into a wound. Vegetative cell multiplies, producing the toxin absorbed into the bloodstream, similar to foodborne botulism, but it's a wound botulism. Unlike food botulism, where there's no fever, wound botulism causes fever but no GI upset. So you don't have the diarrhea with wound botulism. So a distinction there between the two. Okay. We didn't really get to Botox. That is a good question though. What is Botox? What toxin are they putting in your face and lips and forehead, wrinkles, all that? Anybody know?
I don't have
All right, but that is, well, somebody, yeah, we'll start off lab with that question when we get to lab.
I thought it was literally like silicone or something.
Silicone? No,
I
it's a
thought it was
toxin.
substance like
They
that.
call it Botox for a reason.
Botulinum toxin.
Which one?
Botulinum.
Well, it's a different form. It's on a botulinum toxin A is what is in.
I don't know if that's good or bad, do we yet? So yes, we didn't have that in here with our Clostridium botulinum.
It says the toxin was first like discovered by poorly prepared sausages. So
Okay. And you're putting that where?
in your face.
In your wrinkles?
Well, I assume, I don't know.
Okay. In your lips? 'Cause nothing could go wrong with that, right? Nothing can go wrong with doing that. Okay. We're doing good, right? We hadn't had a good lecture like this in a while. We've been usually getting out early the last couple of days. Clostridium perfringens. It is the most common clostridial infection. It gets this morphology in red because I know that's a test question waiting to happen. Gram-positive rod with squared ends like a box car. Y'all know the box car, right? That's like a train car. Y'all seen a train car before? The box car is the one, that has the door in the middle. And I'm just checking to see if y'all actually observe trains anymore. Okay. Heather's laughing because she's... Okay. So being Clostridium perfringens, we have gas gangrene. Okay. This then follows an invasion of necrotic wounds where we have foreign bodies, lack of blood supply. We see this a lot with diabetics. Hemorrhaging into the wound in the presence of other bacterial with lower the redox potential, letting an anaerobe have its way. Proteolytic enzymes of the muscles, hydrolyzed protein releasing amino acids required for further growth of the Clostridia. And there's our boxcar, gram-positive boxcar. And I don't know who gave the nicknames to these, but you will see it as a description of perfringens. Just big old gram-positive rods for me. Oh my gosh, an API strip. Look at that one. Who's calling those oranges positive, right? Y'all are gonna appreciate our microscans today. I hope Thursday you'll feel refreshed and have a little more confidence in your color reading. So I think you'll enjoy that today. Clostridium perfringens, fever, drowsiness, tachycardia. Anybody know what tachycardia is?
That's all right.
Fast heart rate, tender wound with edema, a sweet or foul smelling discharge. How do you go from sweet to foul? But it does. Gas, blisters, the skin may appear gangrenous or erythematosus with a coppery hue. If untreated, this condition can lead to death. And what do they do with those gas gangrene limbs? You might know, toes, limbs, fingers, anything peripheral, right? Cut it off. Surely you've heard that. Like we amputate. Amputate your toes, amputate your foot, come on up to your knee, right? That's our diabetic patient going through that, okay? Definitely. With perfringens, to identify it, we do see a double zone of hemolysis. I was just wondering why we had that hemolytic plate back here, right? Double zone of hemolysis. I think we can see it right in here, right? Almost looks like a bullseye. We have two positive tests that we've never talked about. I think we talked about lipase and indole. I don't think we talked about Naegler's test positive or lactothianase, leaky thianase. We go lessithianase. No help from the audience. No help, but they are positive, so that's two key positives for enzymes, lactothianase and Nagler test. Here's the egg yolk agar again, showing lactothianase production. Egg yolk looks a lot like brain heart infusion, but definitely growing. Lactothianase, opaque white halo. All right, and then we come to, I guess this is our last one of the day, I think. It's C. diff, Clostridioides. Uh-oh, I thought we were with Clostridium. And gosh, there's been a name change, right? We went to, it's like Clostridium-like, so we changed the name to Clostridioides. We're going to talk about that because some people still call it Clostridium difficile. And they didn't want to change it all the way from C because C. diff was so international. Everybody knew what C. diff was. But Clostridioides difficile is the new name for Clostridium difficile that you may hear. So here it is. It's present in our bowels of healthy individuals where carriers or acquired from exogenous sources such as toilets, bedpans, floors, or the hands and stools of hospital personnel. Hands and stools, right? How would, so this is usually called a hospital acquired. Okay, it usually is proceeded with antibiotic treatment over and over and over and over. So the normal flora is lost. So the normal microbiota in your gut, you lose the protectiveness of that because you've taken so many antibiotics in your life or while you were in the hospital for multiple days. And then Clostridioides difficile has a chance to set up. And it likes to produce a toxin too. So intestinal infections with bacterium is the most frequent healthcare linked infection in the US. So the most hospital acquired linked infection in the United States is C. diff. Each year it afflicts more than 500,000 Americans, causes tens of thousands of deaths, and costs the nation's healthcare system an estimated $5 billion a year. The disease, the antibiotic associated pseudomembranous colitis. That's the disease, antibiotics. Then we get a pseudomembranous colitis, inflammation of the colon caused by the antibiotic usage, which disturbs the balance in the colon, presenting C. diff a chance to overgrow the endogenous microbiota, and then it produces cytotoxins once it gets there. Once it starts to grow and set up, we get a cytotoxin production. The antibiotics responsible most often are ampicillin, clindamycin, and cephalosporin. So when we say FYI, I've already warned you that after the next two lectures in an exam, we're going into the antibiotic portion of micro. So this would be definitely something I would want to remember. What is ampicillin? What is clindamycin? What is cephalosporin? Okay, but after that treatment, and most everybody's had an ampicillin taken. Cephalosporins is probably the next most common. What toxin is produced? Toxin A. It's an enterotoxin that is cytopathic in very large amounts. Toxin B is extremely potent cytotoxin. So we get A and B produced from difficile. What are the symptoms? Diarrhea. Okay. And it's usually mucin, fibrin, epithelial inflammatory cell diarrhea. So it's really and it stinks. Okay. You will know if you open up a diarrhea cup, you will know that you've got C. diff potentially, probably because of the smell of it. Okay. This is not going to be a fresh smelling stool sample that you collected. Okay. So what we're going to do, how do we diagnose it other than the diarrhea, is we're going to isolate just about every patient that has diarrhea started while they're in the hospital. We're going to do a C. diff on. That's just the course. That's common. Diagnose, isolate the C. diff from the stool with detection. We can either grow it or we can detect the cytotoxins in the stool. So we've got CLIA wave testing. We can pull off the shelf. All right, take that stool, put it in there, look for the toxin being produced out of that. We can detect that right away, or we can try to isolate it growing it. Using the rapid test, that's the one I'm thinking of. The latex agglutination is probably the quickest way we'll find it. The quickest way we'll pull it out of the fridge. We'll check for the toxin with latex agglutination. What antibiotic will we put the patient on? Okay, we got to treat them. So we're going to put them on metronidazole, vancomycin, and fidaxomicin. Fidamoxin, is that right? Zamycin? How do we say that? Y'all are getting off the hook today. And we still got a little time. Help me with the Fido there. We're going to go fidaxomicin. Is that better? I think that's better. Commonly used to treat C. diff infections, but reoccurrence after initial treatment happens in roughly the third of the cases. This is important because C. diff appears to thrive in people who are normal, healthy gut bacteria have been killed off or diminished. Gastroenterologists recently have begun using fecal microbiota transplantation, FMT, as an alternative to antibiotics for recurrent C. diff. So be careful, because if you have a chronic C. diff infection, y'all know what that is? Anybody who can help me? Who knows? How are we going to use whose fecal microbiota are we implanting? Anybody know? It's not ours. No, not ours, is it? Somebody else's. Yeah, let's just get somebody's, say it this way, somebody's poop, put it in us. A reverse enema. That's what we're doing. Y'all didn't catch that? Y'all never heard of this? I mean, I know you have commercials where they say, oh, take whatever, you see these little ladies on TV talking about taking, this is a reverse. What do they take? What's that stuff called that's in the little green cup? Y'all never seen these commercials to help them get regular? They eat.
Like probiotics.
Probiotics, right? Well, this is a little different. This is using other people's fecal microbiota and implanting it into our GI tract to revive the microbiota to keep C. diff from happening.
How do you do it there?
Reverse enema.
I don't know what that means.
Well, enema soap sud, you go in with soap and you hope you loosen everything up. This would be you put the tube in and you force, instead of soap, you force fecal back up. Some people do this. They think it's very healthy to do this. Anyway, y'all look that one up before lunch too. Okay, Clostridioides difficile, small study in 2013. Heather's having a hard time thinking about it. Found that a single microbiota transplant cleared up C. diff 81% of the time, whereas a standard treatment of antibiotics of vancomycin, right, the biggest of all, we gave vanco, only 31% of the patients cleared C. diff. So the power of microbiota, there we go, 60% better chance of getting rid of it with that than with just vancomycin by itself. There's our gram stain. There it is on the anaerobic blood agar plate. And what is in that that blood agar there? Hemin and vitamin K. Thank you very much. Okay. All right, so we gave you some lunchtime information. I'm going to stop the share. We're going to say bye to Zoom. Now we're looking like, okay, fidaxomicin. I like that, Lauren. Thank you. There's a question on Zoom. Yes. I can't read that one. No. Okay. We'll see you Zoom later today in lab. We're going to end it